Um, Great. I think we have enough people here, so let's get started. First, thanks everyone for joining Dashu virtual seminar. Today, we are very excited to have Dr. Marina Sirota here to speak on this really cool topic, drug discovery in the era of precision medicine. For people who are not familiar with Dashu, we are a nonprofit organization founded to promote research and education in data science. We host scientific conferences, educational seminars, workshops, and short courses. We also provide career development trainings to help junior researchers and scientists. And all of these are creating a global community for people to learn, exchange ideas, and share opportunities. To join our mailing list for notifications on upcoming events, please subscribe from the website. Our membership is free, but we welcome sponsorship or any kind of donation. Even if it's just $1 or $2, it'd be a lot if everyone in the community chips in. It's an investment on your side, and it can help the community grow on our side. We promise that all the money shall be used in place, and we minimize our administrative costs to be less than 3%. For more information, you can go check out our social media pages on WeChat, YouTube, LinkedIn, and Twitter. And here you have the best wishes from our board and volunteers. As volunteers, we do all kinds of things to help run the organization. While enjoying working together, we also help each other grow in career and in life. So if you're interested, you can find the volunteer information on the website, and we welcome you to join the team. And finally, I'm very excited to introduce our speaker today, Dr. Marina Sirota. She's currently an associate professor at Bakar Computational Health Sciences Institute at UCSF. And before joining the team at UCSF, she was the lead research scientist in the Division of Systems Medicine at Stanford University. And she also worked as a senior research scientist at Pfizer, where she worked in drug discovery. And this is very much related to today's topic. And previously, she earned her PhD in biomedical informatics at Stanford. And her research interest is how we can apply computational integrative methods in the context of disease diagnostics and therapeutics, especially how to leverage and integrate different types of omics and clinical data to better understand the immune system. As a young leader in the field, she's been very productive, and she's been recognized as the EMEA Young Investigator Award in 2017, which is an extremely competitive program. And today, Dr. Marina Sirota will speak on drug discovery in the era of precision medicine, in which topic she's a true expert. During the talk, please put your questions and thoughts in the chat box so Marina can answer them at the end of the session. And now I'll stop talking and hand it over to Marina. Thanks, Marina, for joining us. And I look forward to your talk.
Thank you so much. I would like to thank the organizers for inviting me, and I look forward to sharing some of our research with you guys. Well, let me start by sharing my screen. Let's see. Okay, can you see the regular view or the presenter view? Hopefully the regular view.
Yeah, I think it's great. It's
okay, perfect.
It's great now.
Perfect. All right. So I always like to start by saying why I'm excited to be a computational biologist today. And one of those reasons is that there are so many resources that are available to us from the public domain to be able to ask new and exciting questions about disease. And these are just some examples of those resources. The 1,000 Genomes is a reference data set, well, actually has way more than 1,000 genomes that has been sequenced from individuals from different ethnic groups. The Gene Expression Omnibus is a collection of I think over 4 million transcriptomic samples that have been profiled with a variety of different technologies ranging from microarray to RNA-seq to single-cell RNA-seq. The Cancer Genome Atlas is an amazing resource which contains tens of thousands of cancer samples that have been profiled with a variety of omics technologies. And their clinical trials data sets such as IMPORT, as well as drug-related data sets such as the connectivity map and links. And as the cost of sequencing has gone down tremendously in the last 20 years, so has the cost of computation, which really provides an amazing opportunity to integrate these two fields. In addition, people are starting to come up with creative ways to apply some of these technologies. So we've been able to capture and measure genomic and transcriptomic signals for a while, but now we can look at the antibody repertoire, at epigenetics, proteomics, the microbiome, and of course, many of these now on a single cell level. So in my lab, we're interested in asking several different questions. We are interested in using data and data integration to identify new biomarkers. So can we diagnose disease better? To better understand disease mechanism and what might be going on, especially with respect to the role of the immune system. And finally, can we identify new therapeutic strategies? And more specifically in this area of drug repurposing. So can we find new uses for already FDA approved drugs? And this is the topic that I will focus on today. So why would we want to do this? Why would we want to repurpose compounds? And the reasoning and motivation is quite simple. It takes a long time and it's very expensive to take a new drug and bring it to market. And 90% of drugs fail in early development. One way to mitigate that is to identify new uses for drugs that are already FDA approved. This lowers the cost of development and also reduces the risk of failure. And in the last 2 years with the pandemic, we have clearly seen the benefit of drug repurposing and appreciate the ability to take a compound and identify new uses for it, especially in the context of COVID-19. There are many different approaches to computational drug repurposing. There are target-based approaches which stem from genetics, with the idea that if several diseases share genetic markers or genes that might be associated, then maybe they could share therapeutic targets as well. An example of target-based drug development is targeting a gene called PCSK9. And there's a number of drug companies that have been working on looking at that target in the context of hyperlipidemia and lowering lipid levels. There's what I like to refer to is disease-based drug repurposing or phenotypic drug repurposing, with the idea that if certain diseases manifest similarly, either based on clinical manifestations or their molecular profiles, then maybe a drug from one disease can be repurposed for another. And we've seen quite a bit of this type of repurposing done for autoimmune diseases. There are molecular modeling approaches which explore the relationships between drugs and their targets through docking and other kind of structural modeling approaches that have been also quite successful. And there are several companies exploring these options as well. I'll be honest, I'm not an expert in that area at all. And finally, there are network approaches and global signature approaches, where the idea is that one drug, one target, one disease is not sufficient for complex diseases. And this is really the approach that we have taken. So the question that we were interested in asking, and this is actually stemming from my own graduate work at Stanford, was can we use public data to systematically predict relationships between drugs and diseases? So given gene expression data from different compounds, so before and after treatment? What are the expression? How do these drugs modulate expression? And a disease signature. So, sorry, there's already a question in the chat. A recording, sure, I'm okay with it if others are, if the organization is willing to share. So going back to the problem statement, can we use public data to systematically predict relationships between drugs and diseases? So the idea is if you have a disease signature, so individuals who are healthy compared to individuals with a certain condition, can we use the gene expression between the drugs and diseases as a proxy to connect them, to identify some of these potential relationships? So what we really wanted to end up with is a matrix of diseases by drugs, where each cell of that matrix is some sort of a therapeutic score of how likely a given drug is to treat a given disease. So we went out and we looked for data. The disease gene expression data for this first study came from the Gene Expression Omnibus. As I mentioned, this is a public data repository, which contains a large number of samples. I think these numbers are a bit out of date. I think it's actually close to 4, if not over 4 million samples that have been profiled in GEO, and definitely over 100,000 series. And this number of well, here it's 84,000, but I believe it's about 100 or 150,000 right now. These are the individual experiments, many of which deal with disease. On the drug side, we have been using a data set called the connectivity map. This is a collection of expression data of different compounds that have been applied to cultured human cell lines, specifically cancer cell lines. And we'll talk a bit about the benefits and the drawbacks of this approach, but it's a starting point. So the original data set consisted of 164 compounds across almost 500 experiments. So they explored different dosages and different cell lines. And now the expanded data set is about 1,000 drugs across 6,000 experiments. There's even a further extension of this data set called links, where instead of looking genome-wide, the investigators looked at about 1,000 landmark genes. However, we're able to profile tens of thousands of drugs. And of course, you can imagine using these data sets in a way that's complementary, where these approaches look genome-wide at all 20,000 genes, while the links data looks at many more compounds, but only explores expression for about 1,000 genes. So this covers, in terms of the drugs, a broad range of effects, including both FDA-approved drugs and some non-drug bioactive small molecules. So our hypothesis in this work was fairly simple. The idea was if a disease signature is reversed by a drug, then maybe that drug could be used as a therapeutic. We've seen situations where the signatures are similar, and also situations where the signal is mixed. However, again, we focus on this therapeutic hypothesis. So we created a computational pipeline given a disease signature. We query the disease signature against the drug data and come up with a drug disease score. And you can think of the score as a therapeutic, as a correlation, where positive scores are profiles that are similar to the drug exposure, and negative scores are signatures that are opposite or reversal of that drug signature. So we ran this through the disease data that we've gathered, and we gathered data for about 100 diseases in this first iteration. And we're able to identify at least one therapeutic prediction for about half of them. So this is the matrix summarizing those results. You have diseases on the y-axis and drugs on the x-axis, and the negative or potentially therapeutic scores are shown in green. You can see that first of all, here, so black signifies non-significant associations, of which we see quite a bit. However, you do see a number of therapeutic predictions here as well. So we wanted to focus on a single disease and see if we can identify a potential therapeutic for it as a starting point. So we chose to focus on IBD and Crohn's disease. Crohn's disease is an inflammatory disease of the intestine that has an autoimmune component. It affects about 500,000 people in North America, and there's no known pharmaceutical cure. The current solutions are to reduce inflammation with anti-inflammatory drugs and corticosteroids, which have pretty unfortunate side effects. And there are also surgical solutions, which are quite invasive. When we looked at our therapeutic predictions for Crohn's disease, we saw that prednisolone, which is a steroid that is used in the clinic to alleviate some of the symptoms, was one of the top hits. However, there was one other FDA-approved drug that had a better therapeutic score, a reversal score, and that drug was Tipiramate. So Tipiramate is an anti-seizure drug. It suppresses the rapid and excessive firing of neurons at the beginning of a seizure, and it enhances GABA activation. Sort of that's the at least proposed mechanism of action in the context of its original indication. It's used to treat epilepsy and bipolar disorder. It's also an antidepressant, and it has been investigated as a potential treatment for obesity and type 2 diabetes. So we started a collaboration to carry out several studies, animal studies, looking at Tipiramate. The first study was a pilot with a lab at Stanford, with a gastroenterology lab. And then we had 2 follow-up validation studies with 2 independent CROs. So we had here 4 groups of animals, 12, this is a rat model called TNBS, of 12 animals each, including a healthy control group where the disease model is induced that were untreated, where the disease model was induced that were treated with prednisolone, and where the disease model was induced that were treated with Tipiramate. The treatment went on for 7 days, and a number of clinical signs and pathology scores and histology information was collected. We even got endoscopy images on these animals. So looking at these validation data, this is what we observed. Here, we're showing % of animals with clinical signs of diarrhea. And in green, you see healthy animals, so they're doing just fine. In red is where the disease model was induced, and in blue and purple, respectively, is Tipiramate and prednisolone treatment. So we saw a significant reduction in clinical features with both treatments. However, when we started looking at the pathology scores, both the microscopic damage score and the gross pathology score, we saw a significant reduction with Tipiramate in both cases, which we did not observe with prednisolone. And here on the right-hand side, you can see representative histology images as well as endoscopy images on these animals. We further dove into the actual signatures that went into this prediction. And here, the disease signature is on the right and the drug signature is on the left. You can see this is not perfect by any means, but if you squint, you can see that the genes that are downregulated in the disease are upregulated in the drug and the other way around. And when we look at the pathways, we see that a lot of the pathways here make sense. So it's gastrointestinal disease, NF-kappa B signaling, IL-15 signaling, B-cell receptor signaling, inflammatory response, IL-10 signaling. So a lot of immune response pathways. We took a handful of genes here, and these are 2 examples, and validated them in the animal data after treatment. So TRPV1 is an example of a gene that's downregulated in the disease model, and these are genes from the signature on the left, and upregulated by treatment, and IFI30 is upregulated in the disease model and downregulated by treatment. So this was the first application of this approach. This is the method that was developed and the first application to IBD. Since then, we've applied it to a number of different diseases. Dermatomyositis is an autoimmune disease of the skin. There's been a number of applications in cancer, and Bin Chen, who invited me to give this presentation, has led a lot of the efforts of drug repurposing in cancer. It's a natural application of this approach. Because the drugs are tested in cancer cell lines, and we can do extensive matching to be able to match the models in which the drugs are tested to the actual human data. We've looked at pregnancy outcomes like preterm birth, and we're able to identify lansoprazole, which is an antacid for potential as a potential compound that might reduce the risk of preterm birth. And more recently, we've been looking into applications of this approach to neurodegeneration and Alzheimer's disease. And that's the story that I would like to focus on next. So this is a manuscript that was published last year. The work was led by Ali Taubes, who was a graduate student jointly in my lab and Dr. Yidong Huang's lab. And this is a collaborative effort with many different individuals. All the animal work was done in Yidong Huang's lab. And then we collaborated with Ben Glicksberg at Mount Sinai for some validation efforts, as well as Ben and many, many others. So the idea behind this work was to apply a precision medicine approach to drug repurposing. So the question that we were interested in asking was, is Alzheimer's disease different if we stratify patients by their ApoE genotype? As you might know, ApoE is one of the biggest, or actually the biggest risk factor for Alzheimer's disease. And individuals with ApoE44 genotype have a much stronger risk for Alzheimer's and also are more likely to develop it early. So what we wanted to do is we wanted to take expression data, and instead of generating a single signature for Alzheimer's, stratify the signatures by ApoE genotype. So this was the data set that we looked at. This is looking at the samples themselves, and you can appreciate the different distributions by genotype. So ApoE33, 34, and 44. This is what the PCA of the data, the expression data looks like, looking at case control. So you can see a little bit of the separation. And then this is looking at the same data stratified or colored by genotype. So you can see that there are some differences here as well. When we looked at the signatures and the pathways, we saw that there were some genes that were overlapping across the three signatures. So ApoE44 shown in red, ApoE34 shown in gray, and ApoE33 shown in blue. However, there were quite a few genes that were specific to each genotype. And you can observe this even more starkly on the pathway level, with a large number of pathways specific to ApoE33 and ApoE44, while the signal for ApoE34 was a bit mixed. So then we went on to, we went on to carry out the drug repurposing predictions for the three genotype specific signatures, as well as the general signature of all the samples combined. And again, we were interested in focusing on ApoE44 disease. And what we observed here, that a drug called bumetanide came out as highly significantly reversing the expression signature for ApoE44 disease, while it still had negative reversal score for the other signatures, as well as the general one. It was really one of the strongest hits for ApoE44 disease. And here on the right-hand side, we're showing different ways to look at that signature reversal. So here you can see the disease signature for ApoE44, and you can see again the genes that are upregulated in the disease are downregulated in the drug, and the other way around. And this is showing the same shift as a histogram. So what is bumetanide? Bumetanide is a loop diuretic that works by decreasing the reabsorption of sodium by the kidneys, and it's used to treat edema and hypertension. We wanted to test this in an animal model to look at behavior. And to do that, we carried out the water maze test. And this is what that test entails. There is a pool of water, and I'm not a mouse model expert, but as I understand, mice don't like to be in the water very much. There is a hidden platform in one of the quadrants, and mice are entered there. They enter this pool in one of the quadrants, and they look for the target quadrant. So what we're looking at here is the time that it takes for the animals to escape into the target quadrant. So here you can see several data points. Here in black, you see the 3-3 disease. So both the untreated animals as well as animals that were treated with high doses of bumetanide, and you can see that the curves overlap. There's no phenotype observed in the 3-3 animals. In the 4-4 animals, however, the solid line represents just the individuals, and the dashed line represents the animals that were treated with bumetanide. And you can see a significant reduction in the escape latency. So these animals are looking closer to the ApoE 3-3 individuals at 24 months of age. This was very promising. And we wanted to see, and we've also, I don't have time to go through all the data now, but we've done extensive work looking at single cell characterizations, both in the treated animal models, as well as looking at human neuron data treated with bumetanide to confirm reversal across many of these data sets. But what I wanted to focus on here today is more of the computational work. And the question that we wanted to ask following some of these promising animal data is, can we further validate these findings using real world data in the electronic medical record? So the electronic medical record provides an additional amazing opportunity to start looking into real human information on real human patients. So this is data that is recorded when you go to see a doctor, and the UCSF electronic medical record spans from about 2012 to today, so almost 10 years. It contains the data that was used for this manuscript, containing about 1.3 million patients, and included information on allergies, diagnoses, encounters, immunizations, lab test results, medications, procedure orders, vitals, and we even have imaging information. So it's quite extensive. So this is what the data look like. And you can clearly see the kind of diverse population, roughly equally split between males and females across different ethnic groups, and as well as the age distributions at UCSF. There are incredible efforts. So Atul Butte, who leads the Baker Institute for Computational Health Sciences at UCSF, has been working to aggregate clinical data across the whole UC system. And we now have access to all of that. And this number is a little bit out of date. It's about 20 million patients, which is quite incredible. And it's all de-identified and available for research purposes. It's important to note that these data originally are collected for billing. So they're not perfect, but the fact that we have data on so many individuals is quite promising. And I will show you how we've been using it in the lab. So here, we wanted to take the data, we started with UCSF, and do what we call kind of a retrospective in silico clinical trial. And we wanted to ask, from the individuals who are on bumetanide already, as well as matched controls, what are the chances, or what is the prevalence of Alzheimer's disease in these groups? So we started with 1.3 million individuals. We focused on individuals who have medication information, and about 5,500 people were exposed to bumetanide. Not all of them, again, the data is messy, not all of them had demographic information, and only about 1,800 were over the age of 65, which is the age group that we're interested in terms of looking at the risk of Alzheimer's. We carried out propensity score matching using a ratio of 1 to 2 of cases to controls, matched on age, sex, race, and ethnicity, as well as the indication for bumetanide, which is hypertension and edema. And again, we wanted to see what is the ratio of Alzheimer's versus individuals who didn't get Alzheimer's, at least at that stage of their life. And we did this independently at UCSF and also replicated in an independent electronic medical record in collaboration with Ben Glicksberg and his team at Mount Sinai. So this is what the data looks like at UCSF. You can see that the prevalence of Alzheimer's is significantly lower in individuals who are on bumetanide in comparison to the matched controls across all 10 iterations. We see even a higher difference in the Mount Sinai data. So again, this is strongly significant. There's more individuals here. So the starting point was 3.9 million. 800,000 had a medication record, 3,000 exposed to bumetanide, and 2,400 almost were over the age of 65. So this was incredibly promising to us, and we've been working since to explore the data in the electronic medical record further. This is work by Alice Tang, who's a very talented MD PhD student currently in my lab, and it was just published last month in Nature Communications. The idea here is to look at the heterogeneity of the Alzheimer's disease population in the UCSF electronic medical record. And again, the data that went into here was demographics, diagnosis codes, medications, lab test results. The Alzheimer's cohort is almost 9,000 individuals, and we carried out a matched control cohort of about 1,700 individuals, so 1 to 2 matching like before, based on demographics. We visualized the data, and I'll show you some of that in a second. We looked at comorbidities as well as lab test results and medications. We further validated what we have observed in the Mount Sinai cohort. So these are principal component plots where each dot is a patient, both at UCSF on the left and Mount Sinai on the right. These are based on comorbidities, so diagnosis codes, and excluding anything relating to Alzheimer's. And this shows that despite the fact that we're excluding Alzheimer's disease as a diagnosis, you can see a clear separation between individuals with Alzheimer's, shown in red, versus healthy controls, shown in blue. And this is consistent across both cohorts. We've built networks looking at comorbidities in individuals with Alzheimer's in comparison to healthy controls. And you can see that there are many more relationships across different other diseases, including hypertension, urinary tract infection, as well as others in the disease cohort as opposed to the control cohort. We are able to look at diagnoses that are enriched in Alzheimer's in comparison to controls. Obviously, some of these make a lot of sense, like dementia and mild cognitive impairment, but there are others that are interesting, such as urinary tract infection, hypertension, hyperlipidemia. There are some diagnoses that are enriched in controls, including several cancers, which is quite interesting. And again, we were able to replicate about 80% of these associations in the Mount Sinai data. So this is showing a log plot between the UCSF data on the x-axis and Mount Sinai data on the y-axis. We've also carried out analysis looking at sex-specific associations. So you can see a Miami plot where the associations in women are shown at the top and associations in men are shown on the bottom. And you can see that many of the conditions that we've seen, such as depression, hypertension, osteoporosis, and UTI, are enriched in women and not necessarily to the same degree in men. You can see other conditions that are enriched in men, and I apologize, the color here is a little bit bleak, but things like sleep apnea, irritability, and Parkinsonism are enriched in males with Alzheimer's and not necessarily in women, which really points to the fact that the disease might be different, not only if we stratify by ApoE, the genetic marker, which is what we've looked at molecularly before, but also based on sex. And again, many of these replicate in the Mount Sinai data, which is shown by these graphs on the right. Finally, we've been looking at lab test results, and this is showing the data both at Mount Sinai and at UCSF, and allows us to identify lab tests that might be associated with with Alzheimer's disease in males versus Alzheimer's disease in females. Certain lab test results are consistent, while others, such as this group here, are specific to females or specific to males. Finally, we've carried out drug association analysis across many of the compounds, and are able to identify a number that are enriched in individuals with Alzheimer's, and also some that are enriched in healthy controls, and we're investigating those further. One interesting one is furosemide, which is also a diuretic similar to bumetanide, and again, we're investigating these in further detail. So I guess on our side, what's next? We are starting to look into single cell data and single cell type specific approaches. This is a manuscript that's under review, looking at stratifying by ApoE genotype, both across using several single cell data sets. And we are working on ways to extend the drug repurposing approaches to single cell data. Here we've, in this paper, we've looked at cell type specific ApoE stratified signatures across individual cell types. And we see that, yes, there are a lot of signatures that are in common. So these are log-log plots across the different cell types with ApoE on ApoE33 on one axis and ApoE 44 on the other. However, there are some signals that are specific. So I guess summarizing our Alzheimer's disease efforts, we've established an ApoE genotype dependent transcriptomic signature of Alzheimer's using publicly available data. We've done this in the bulk, for the bulk existing data, but are also extending to single cell. We've identified bumetanide as a potential candidate for ApoE4 specific disease. We've shown that treatment with bumetanide reduced AD-like cognitive and electrophysiological deficits in mice, and have been leveraging single nucleus sequencing to explore the effects post-treatment in mice and human cells. We've also used electronic medical records to show lower prevalence of Alzheimer's associated with bumetanide treatment using real-world data. And we're extending both the molecular approaches as well as the electronic medical record analysis further, as I've shown you. So in the last few minutes, I wanted to shift gears and tell you how we've been using these types of approaches to identify new therapeutic compounds for COVID-19. So everybody knows the ongoing pandemic, and this is as of October 2020, there were over 255 million cases of COVID-19, and of course now many, many more. There are a few effective treatments that have been discovered. However, as you are aware, new drugs have a long development timeline, and here really drug repurposing has come to the forefront. And many groups have been working on approaches to carry out this type of work. We ourselves asked what we could do when the pandemic started, and this has really been a collaboration with almost everybody who was part of my lab at the time. The work was jointly led by Brian Lee, Guy Androletti, and Timiko Oskotsky in the lab, but again, many, many folks in the lab have collaborated, and it was published in Scientific Reports last year and presented at ASHG during the plenary session in 2020. So we started this actually in April of 2020. And at that time, there were only a few transcriptomic data sets that were available at the time. And these were the data that we had. These are the signatures that we had. There were three of them. One came from human adenocarcinomic alveolar basal epithelial cells infected with SARS-CoV-2, contained about 120 genes, and enriched pathways that were observed included cellular response to virus infection, not surprising, and humoral immune response. There was a data set of organoids grown from gut tissue, where the gene expression signature was 125 genes. And then this was the first COVID-19 actual patient data set, which was incredibly limited, 2 patients and 3 controls. And this is RNA-seq data, and the signature was much stronger. It was almost 1,500 genes that were identified here. So looking at the overlaps across these signatures on the gene level, we actually did not observe a huge overlap. But looking at the pathways, we saw quite a bit that was in common. So pathways that were upregulated included interferon response, E2F targets, oxidative phosphorylation, and ones that were downregulated included adipogenesis, mTORC1 signaling. And the pathway analysis was done by Catherine Yu in my lab. So we wanted to see what was the overlap in the therapeutic predictions. And these are showing top 15 hits for each signature. And so this is the alveolar signature, this is the organoid signature, as well as the patient signature. And here we're highlighting several compounds in green and purple. The purple ones are ones that are in common across these signatures. And several of them made sense to us at the time. We saw that things like haloperidol and valproic acid were also identified independently through a proteomics approach by another group at UCSF, and colchicine was at that time in a clinical trial. So we thought these results were quite promising. We wanted to focus on the drugs that were common across these signatures. So this is summarizing, this is a set of 25 hits that were common to at least 2 signatures, both visualized as a heat map on the left, as well as a network on the right. And in this network, we're showing not only the compounds, but their drug targets, known drug targets from drug bank. And if the drug targets are shared, then they're highlighted in yellow. So you can see that there are some shared drug targets across some of these compounds. So there were 4 drugs that reverse all 3 signatures, and these were the 4. Becampicillin has been used to treat bacterial infection. Clofazimine is an antimycobacterial used to treat leprosy. Haloperidol is a psychiatric medication used to treat schizophrenia. We know that it can bind to sigma-1 and sigma-2 receptors, and it has been found to decrease viral growth in SARS-CoV-2 infected Vero6 cells, as was shown previously. And valproic acid has been used to treat epilepsy, and it has been implicated in inhibition of HDAC2. So we wanted to validate some of these drugs, and we partnered with 2 independent groups. One was a group by Melanie Ott. So this work has been carried out by Albert Gracia. And the idea here was to look at infection and cell lines infected with the virus and see what would be the viral load. And you can see that here is an example of treatment with remdesivir as well as treatment with haloperidol. And in both cases, you see a significant reduction, which is also shown by this curve here. We furthermore collaborated with Adolfo Garcia Sastre and validated, they have a high throughput system to validate a number of these compounds. And here we're showing some of these results. So the black line is cell viability and the red line is viral load. So we want the black line to stay as flat as possible, while the red line would like to go down. And you can see that for a number of the compounds that we looked at from the prediction, we saw those trends exactly. So in summary, we applied our computational drug repurposing pipeline to a number of different diseases, including COVID-19. Here we identified 25 common hits, which are shared across at least 2 signatures and 4 consensus hits. The validation work was carried out by 2 independent groups in 2 human cell lines and showed viral inhibition by 11 out of 16 common drugs and 3 out of 4 consensus drug hits. So again, this work has been published last year. And since then, we've been mostly leveraging electronic medical records to look at different types of hypotheses for COVID-19. We've had the opportunity to get access to an incredible resource by the Cerner group. And what they have done is they've aggregated clinical data across 87 institutions across the US that includes and made the data for COVID-19 specifically available to researchers. This includes over 500,000 emergency room and hospitalized patients diagnosed with COVID-19, some of them confirmed, some unconfirmed. And the data that we have access to include demographics, medications, procedures, encounters, diagnoses, lab test results, imaging data, as well as past medical history for up to 5 years for some of the individuals. We had a few hypotheses that we wanted to test. The first one was looking at statins and COVID-19. This is work led jointly by Ivana Marek from Stanford and Tomiko Oskotsky in my lab and was published in Frontiers Medicine in 2021. We know that statins have an anti-inflammatory and immunomodulatory properties. And we partnered with, as I mentioned, a group at Stanford led by David Stevenson to look at whether in patients with COVID-19, we could observe reduced disease severity and reduced mortality rate in patients who are already on statins. So we applied a similar approach to what we've done with bumetanide, looking at 10 independent iterations of individuals on statins, of which there were almost 3,000 and matched controls. And we looked at mortality rate. And we saw consistently across the 10 iterations, the patients who were on statins had a lower mortality rate than individuals who were not. The reduction wasn't huge. You can look at about 16% to about 20%, but nonetheless, it's important to note. As I mentioned, that work was published previously, and then at the end of last year, we also published another retrospective study specifically focusing on SSRIs. The study design was very similar. The work here was again jointly led by Tomiko Oskotsky and Ivana Marek with collaborators both at Stanford and UCSF. So this is what that framework looked like. We started with almost 500,000 patients, as I mentioned. About a little bit over 100,000 of them have laboratory tests confirmed or ICD-10 confirmed COVID diagnosis. Some of them, there was of course some missing data. A bit over almost 3,500 individuals were exposed to SSRIs during the defined time frame. And again, we looked at a repetitive propensity score matching across the board, looking at individuals who were not exposed. We further stratified into different types of SSRIs, looking at fluoxetine or fluoxamine or other SSRIs. And here, what we observed was that overall, we saw about 8% reduction in mortality rate among patients with COVID-19 who were on SSRIs in comparison to matched controls, and a larger reduction, almost a third, in the patients who were treated with fluoxetine or fluoxamine, which is a subtype of SSRIs. We wanted to, did we want to look at SSRIs? There has been a link between serotonin and the immune system. People have shown that fluoxetine can decrease levels of the various cytokines, such as IL-6, and might modulate the sigma-1 receptor and the receptor ERA pathway, thereby reducing damaging aspects of the inflammatory response. These are just suggestions and speculations from the literature. This type of analysis only allows us to identify an association. And of course, a lot more work, both on the clinical side, validating this further, but also on the biological side, understanding the disease mechanism, need to be done. This work has been picked up by the press quite a bit, so we're hoping that it can make an impact in the overall community and lead to further studies. So in terms of our ongoing work, we have been applying computational approaches using both molecular and clinical data for drug discovery. We're currently exploring combination therapy approaches, as well as applying machine learning, getting into single cell measurements and ways to incorporate that into drug prediction, and furthermore, enhancing our approaches with electronic medical records. And the hope is that by combining the molecular and the clinical data, we can maybe get a little bit closer to precision medicine. With that, I'd like to thank everybody in my group, those who have contributed to many of the projects that I've discussed today. Our collaborators at the Gladstone Institute, so Yedong Huang and his team on the Alzheimer's disease work, many folks at Stanford, including Atul Butte and Joel Dudley, as well as others in the original method development, Melanie Ott and folks at Mount Sinai, Adolfo Garcia Sastra on the COVID-19 validation work that I presented, as well as Ben on computational analysis of various projects that I've mentioned. And with that, this was our team. This was us in February 2020, I guess, almost to the date 2 years ago. And we've been remote on Zoom for the past 2 years with small occasions to get together. And with that, I'd like to thank everybody for your attention. And I would love to take any questions.
Thank you, Marina, for this excellent talk. And it also seems the perfect timing. Marina, can you see the questions in the chat box, or do you need?
Yes, I do. I can see. Okay, so do you consider ApoE 22, 23, and 24? We have not yet because the numbers are very small for those ApoE genotypes. But as we get more and more data, we would like to include those as well, of course. What is the major obstacle for pushing your repurposed drug findings to further pharma development and clinical trials? Are pharma companies interested in repurposing existing drugs? This is a great question, and having worked in pharma for a bit myself, I will say it's challenging. We file IP on our predictions, but often it's hard to, especially for things that are off-label or off-patent, it's hard to justify financially moving forward. However, we've seen that when there is a need, for instance, in the example of COVID-19, these efforts do move forward, and they can move forward pretty quickly. So my hope is that these examples will pave way for future repurposing studies and enable them moving forward. Did your team have a change to see the efficacy of combinations of repurposed and conventional drugs? This is a great question. It's something that we're really interested in looking at moving forward. For Alzheimer's, I will say there is no good existing treatment. So in certain cases, there isn't kind of a ground truth to compare to. So in that case, we haven't explored. For COVID-19, I think it would be really interesting to see what would be the combination of, let's say, remdesivir and some of the compounds that we've predicted. We haven't looked at that, but that's a great point. Could you explain how you cross-reference connectivity map data to geo-expression data? What is the algorithm that you use to build the drug gene score? So this is a non-parametric Kolmogorov-Smirnov-based algorithm. And I will say you can think of it as a correlation. The idea is you look at a gene signature and genes that are upregulated in the disease and downregulated in the disease. And then we look at the ranks of those genes in the drug data. And we ask whether the genes that are upregulated in the disease are downregulated in the drug, and whether the genes that are downregulated in the disease are upregulated in the drug. And that's what the score is based on. Yeah, Jenny, please reach out. I would be happy to work together and figure out how to bring some of these data science approaches and explore. Absolutely. Other questions?
Yeah, it seems we still have a few minutes. So anyone, if you have questions, feel free to unmute yourself. Looks like there's a new question in the chat box.
Do you plan to include the links data set as well? Great question. So we've had limited success with the links data set because the 1,000 genes is quite limiting to the disease signature. And the imputed data across the whole genome in our hands has been quite limited. So we're thinking of ways to include it, but we haven't yet. Other questions? Data beyond transcriptomic data, like epigenetics and SNPs. That's a great question as well. I think on the disease side, we've integrated expression with other molecular measurements. But in terms of the drug information, unfortunately, we don't have, I would love to have an epigenetics data set. I have a wish list. I would love to have epigenetics or proteomics data on the drugs. It just doesn't exist yet. I would love to have drug data on relevant cell types, because all of these drug data to date have been on cancer cell lines. So there's a lot that we would like to do further, but we just don't have the data on the drug side yet. The disease side has been profiled extensively. So I'm hoping folks, and maybe we will generate additional data sets that will enable those approaches. Are you aware of any drug repurposing application with protein antibody drugs? Absolutely. So you can imagine for specific targets to look at whether certain targets are common across other diseases and move forward with that. It wouldn't be a transcriptomics-based repurposing approach, but you could look at specific targets across disease expression data or disease proteomics data and see if you can repurpose drugs that way. Alternatively, if you had expression data of tissue or cells treated with antibodies, you could use this type of repurposing, kind of signature-based repurposing approach for protein and antibody drugs as well. So there are opportunities to do that. Other questions?
I think, is there anyone speaking right now? No? I can allow one more question since it's about time. Okay, I think that's it for today. Thanks, Marina, for this excellent talk. This topic is so interesting and promising, and I I just also thank everyone for joining our Dashu virtual seminar and hope to see you next month in our seminar.
Excellent. Thank you so much for inviting me. It was my pleasure. Nice to meet everyone.
Sure. Hope to invite you again in the future.
Absolutely. Thank you. Take care.
Thank you, Marina. Bye.
Bye.
Take care.